Good morning, everyone. Welcome to the lecture. Welcome to the another lecture of this particular subject, that is basics of virology. So today, what we are going to discuss today, we will be focusing on a very important aspect in virology, and that aspect is oncogenic viruses. Now, we all know that viruses are very potent pathogens, very potent human pathogens as well as animal and plant pathogens. But in certain cases, it has been observed that the viral infection or the persistent or chronic viral infection has been attributed to be the root cause of certain types of cancers. So, what we are going to discuss today? See, today we will be focusing on those viruses which have been reported to cause cancer, and we will be discussing the mechanism of carcinogenesis induced by these viruses in detail. What are the proteins actually that are being targeted by this particular viruses, or what are the signaling pathways that are otherwise are able to maintain the normal physiological response or normal cellular response, but then in response to these viral proteins, those critical proteins are getting aberrant in function, and then their aberration or the impairment in their function is inducing cancer. So in nutshell, this would be our main object, that we should be able to understand the detailed mechanism how a virus is able to induce cancer, and what are those viruses, and simultaneously we should have a very clear understanding by taking the normal functioning of a cell in hand to hand. We will be comparing how virus induces the aberration in the normal cellular balance, and what are those critical proteins which we call them as tumor suppressor genes. So we'll be talking about the tumor suppressor genes also. So by the end of the lecture, I hope that you will be getting the complete gist that how this particular mechanism is happening, and what are tumor suppressor genes, what is the interplay or the cross connection between the viral proteins, the virus infection, and the tumor suppressor genes, and genesis of cancer. So let us begin the discussion by first knowing what are those kind of viruses. What are those viruses? If we are talking about the oncogenic viruses, then the human oncogenic viruses can be categorized into two parts. See, viruses have their own genome. The genome varies, whether it can be a DNA, it can be an RNA. We had discussed this in previous multiple lectures, that viruses may have double strand DNA, they may have single strand DNA, they may have double strand RNA, and they may have single strand RNA. They may have multiple copies of single strand RNA, making them diploid, and certain cases triploid also. So the family classification is very large, and genomic classification is very large. That's why Baltimore classified them in 7 different classes, right? And based on those 7 different classes, we study the complete viral classification. So among those, when we are talking about the DNA viruses, and the DNA viruses that are known to cause tumor, so the small DNA tumor viruses are, like for example, our papilloma virus, which is again a very potent virus, which has been shown in oropharyngeal cancer. See, human papilloma virus (HPV), we will be taking its example, and we will be studying how this particular virus induces cancer. Then SC40, adenovirus, these all three viruses have been known to induce tumor. Then when we're talking about the large viruses like herpes virus, the herpes virus again we'll be discussing, and that induces the Kaposi sarcoma, as well as the Epstein-Barr virus. Not even that, the HIV patients, the HIV condition, the cases, HIV cases have been the classical condition that determines that yes, the HIV infection might be there is Kaposi sarcoma. Then other viruses like hepatitis hepatitis B virus is also known to be to induce cancer. Then when we are talking about the RNA containing viruses, we are talking about HTLV, the leukemia virus, the retrovirus, that is HTLV, it induces T cell leukemia. Whereas when we are talking about the other virus, that is HCV, hepatitis virus CH. So see, these are certain examples of DNA viruses. If we talk about what kind of cancer or what cancer type these viruses are inducing, so we have to understand, and we have to first remember that if we are talking about the papilloma virus, what kind of cancer type, what kind of tissue type that these viruses will be targeting and inducing the carcinosis. So when we are talking about the papilloma virus, it affects the warts, the cervical carcinoma, or the genital carcinoma. When we are talking about the ABV, Epstein-Barr virus, it's known to induce Burkitt's lymphoma. Then when we are talking about the herpes virus, the 8 variant Kaposi sarcoma, whereas the hepatitis B virus, HPV, hepatitis, the name itself suggests the hepatocellular carcinoma. When we are talking about HBLV, herpes simplex virus, then we are talking about the lymphomas, B cell lymphomas. So you see these DNA viruses are the classical example which are related to the development of human neoplasia. And when we are talking about development of cancer, then the tissue type what we have discussed in this particular slide that needs to be remembered. Then when we are talking about the RNA viruses, see, when we are talking about the RNA viruses, then we have to remember that we are talking about three specific viruses, that is HTLV1, HTLV2 and HIV. See, HTLV, the name itself is saying human T cell leukemia virus. So it induces T cell leukemia or lymphoma. Then the second variant is inducing hairy cell leukemia. And the HIV, see the terminal cases of HIV are diagnosed with lymphoma itself and sometimes with Kaposi sarcoma. So these association between the viral infection and the cancer have been demonstrated in multiple conditions. So what happens that when you are seeing that an individual has developed a cancer, and then when you are determined to identify the origin of that particular cancer, then you come across the patient history and you see that there was a persistent infection of a certain kind of virus which has led to the transformation of a normal cell to a cancer cell which grows rapidly with this uncontrolled cellular proliferation. So that means the viral protein or the persistent viral infection has done something, has induced a change in certain regulatory factors. Those regulatory factors were responsible actually for maintaining the normal cellular proliferation. So that proliferation is now not being regulated. And that, in a nutshell, is the mechanistic approach adopted by these viruses to induce cancer, and that we'll be discussing. So see, before going ahead to in detail mechanism, let us just brush up how these viruses go for their replication mode inside the host cell. So when we are talking about two major classes, this is DNA and RNA, and when we are talking about the DNA viral genome, so what is happening? The DNA viral genome inside the cytoplasm gets replicated by DNA dependent DNA polymerase. It can be both from the host or the virus can code by themselves. Then in order to express the viral protein, these viruses require host machinery, and the host machinery, by host machinery I mean all the raw material that is required for the synthesis of mRNA as well as the enzyme that is RNA polymerase. And then by the flow of genetic information, that is central dogma from DNA to viral mRNA and then viral mRNA to viral protein, and that is how these tumor viruses are producing their proteins. And when we are talking about the RNA viruses, they have the RNA genome, and these viruses also encode for reverse transcriptase. So the reverse transcriptase will be performing its function and making the DNA from this particular RNA genome, and that DNA is again getting replicated with DNA dependent DNA polymerase. And if this RNA virus wants to replicate its number, then it replicates its number by using DNA dependent RNA polymerase, which is a normal RNA polymerase, which gives rise to multiple copies of the RNA, which can be packaged and can be released to infect the other cells. And then when we are talking about the processing part, when this mRNA is being made and when this mRNA is used to produce the viral proteins, then in between the mRNA which is formed goes for processing by using host machinery. It can go for splicing by using the host spliceosomes. So you see what RNA viruses are doing. RNA viruses are maintaining their number by the enzyme which they are coding, and then they are utilizing the mRNA, and that mRNA is going ahead to form the protein. So this is just to brush up that yes, we have already understood the replication cycle of multiple viruses in detail. So it's just to brush up a general phenomena how these two categories of viruses are replicating inside the host cell. Of course, this flowchart doesn't summarize the technical aspect of each and every virus, which is very peculiar. The genome, what they encompass, and the proteins, what they code, they perform different functions. But yes, in the general umbrella, this flowchart fits right in. So when we are talking about the association between the virus and the cancers, see, it has been shown that the cancers worldwide, and among the prevalence of all the cancers, almost 10% of them have been shown to be caused by viral infections. And in fact, greater than 85% among that majority is happening in the developing countries. and the developed countries see very few cases of these particular virus, mainly because of the pathogenic conditions or the conditions which are favorable for transmission of those particular virus. That is the main thing. Now when we are talking about the mechanistic part, then broadly there are two mechanisms that can lead to viral induced cancer or viral induced carcinogenesis. First is indirect and the other one is direct. So let us talk about indirect. We say indirect mechanism in terms that the cell which is getting infected by the virus is not actually developing into a tumor cell, but the cell which was not getting infected, which was not affected, is now transformed because of the viral infection. So you see, the cell that give rise to malignant tumor, they have never been infected by the viruses. So what exactly is happening? See, when the viral has infected, the immune response will be generated. The host immune response will be generated. It will try to eliminate that virus. So what is happening? Suppose there is a persistent presence of virus. So persistent immune response will be generated, right? If the viral infection lasts for let's say 3 months, then for continuous 3 months, the host body is generating the immune response. And what are those immune responses? All those immune responses comes in the form of inflammation. Then that's the case of developing chronic inflammation. And that chronic inflammation leads to tissue damage or regeneration or transformation of certain proteins, which can lead to excess of cellular proliferation, loss of checkpoints, and that might lead to uncontrolled growth, which can lead to cancer. And that is how inflammation is the root cause of majority of the cancer. Inflammation, the chronic inflammation, not the acute one, the chronic inflammation is the root cause of multiple disorders, and cancer is one of the inflammation mediated disorder. So see, indirect mechanism, when we're talking about, the virus is inducing the inflammation for a longer period of time, and that inflammation that is being induced is may result in cancer. So in nutshell, if we talk about even in the indirect or direct mechanism, what we are talking about since past few minutes is uncontrolled growth. That is how we are defining the cancer, right? What is uncontrolled growth? What is the meaning of that uncontrolled? See, in normal cell, there is a phenomena called a cell cycle. There are defined phases, there are defined functions in that phases which the cell is performing, and there is a defined time period for which that particular cell is there in that phase, whether it is G1, S, G2, M, what, and you can pick any phase, and there's a definite time period in which the complete function of that phase will be performed. So when we are talking about uncontrolled growth, that means there is no regulation on that particular cycle. There is no regulation on that particular cell cycle, right? And then the proteins or the factors that are responsible for regulating those cells, that means in cancer somehow those regulators are getting aberrant. And what are those regulators? What is the name that is given to those regulators? The name that is being given to those regulators is checkpoints. If the checkpoints which are present inside the cell cycle, they gets mutated or they get impaired, then there will be no one to check whether any damage in the cell is getting propagated ahead or not. Because under normal condition, if there is any kind of DNA damage, then the checkpoints stops the cell cycle. It puts either in senescence, it puts that particular damaged cell towards apoptosis or other pathways, thereby giving ample time for the cell to repair that damage. And if repair cannot be performed, then the cell is removed by apoptosis. But if the checkpoints are not performing correctly, then what will happen? It's very simple, right? It's very simple. What will happen? There is no checkpoint. Whatever the damage is there in the DNA, nobody is there to check it. Let's go ahead and perform the cell cycle. Let's form a cell which gets mutated, which has no control over its growth, and then that particular cell will give rise to multiple copies of that cell, and that's how the tumor is getting developed. So the basic mechanism is just the failure of growth inhibition. The failure of growth inhibition. growth inhibition or uncontrolled growth, or there is failure in checking the cell cycle. That is the main function. And what, in terms of the technical language, what we are calling that, see, the genes which are actually responsible for proliferating the cells, we are calling them as oncogenes. Why? Because oncogenes are there, genes which are responsible for oncogenesis, right? So the genes which are actually causing the proliferation of the cell, but uncontrolled proliferation of the cell, those genes are called as oncogenes. Then there are opposite side. Then on the opposite side, there are genes which are called as tumor suppressor genes. Although in later stages I'll tell you that that's a misnomer, but the function of tumor suppressor gene is to apply the brakes on what? On cell proliferation. These are the checkpoints. Tumor suppressor genes are the checkpoints. Will check the function of the oncogenes. They will say, no, you cannot go for uncontrolled growth. There has to be a controlled cell proliferation in order to go for normal cellular responses. And these tumor suppressor proteins, or the genes, products of these tumor suppressor genes, forms a coordinated network called as checkpoints. For example, the two classical checkpoints, the two classical proteins which have been very deeply investigated throughout the oncogenesis part or the oncology part, or different functions, whether it is apoptosis, whether it is cell senescence, whether it is autophagy, whether it is tumor microenvironment response. So these proteins, retinoblastoma and P53, perhaps one of my favorite proteins, P53. These have been investigated thoroughly in terms of their role as proteins in the regulatory network, the regulatory functions, which is able to recognize, they act as sensor to recognize any kind of genotoxic stress, whether there is a DNA damage, then they will be activated, they will form a coordinated networking, thereby shutting down the proliferation, giving the cell ample time. So you see these two proteins up, there are many other proteins also, but we are talking about, we have taken the example of retinoblastoma and P53. So you see these two proteins are so much crucial in order to have a very normal cell cycle. So when I'm talking about the cell cycle, what I'm talking about, I'm talking about G1 to S to G2 to M transition. And when we are talking about checkpoints, we are talking about the G1 to S checkpoint, G2 to M checkpoint, and the cyclin molecules along with the cyclin dependent kinase molecules. So when we are talking about cyclin D with CDK4 or CDK6 to 1, these are the proteins which are responsible for a cell to transit from one state to other. So when we are talking about the transition of from G1 to S phase, then cyclin D along with cyclin dependent kinase 4 is responsible for the transition, and the retinoblastoma is also present here to check out, to as a checkpoint, to check whether there is a damage or not. If there is no damage, then retinoblastoma will get phosphorylated and it will allow the cell to pass from G1 to S. And the phosphorylation of this retinoblastoma is performed by what? A kinase enzyme, that is cyclin dependent kinase. This cyclin dependent kinase 4 will phosphorylate and go for the transition of the cell. Then there are certain inhibitors also. If there is P16, 15, 18 and 19, these are CDK inhibitors in this particular state. When we are talking about CDK6 or CDK2, there are other certain sets of inhibitors. So you see the complete network is very well coordinated. Why it is coordinated? Simply to prevent the uncontrolled growth. There should be a controlled cellular proliferation. So when we are talking about the oncogene expression, what? In normal cells, the cells which are actually normal, they are not infected by any kind of viruses, there is no mutation. If you see that there is at a certain point, when there is an expression of these oncogenes, what will happen? The cells will go to quiescence, or they will go to permanent cell cycle arrest, or they will never be allowed to go for uncontrolled proliferation. Why? Because these tumor suppressor genes or checkpoints are very active. This immediately saw that there is a DNA damage. We should not allow this cell to go ahead. We have to put it into a senescent state. So a permanent cell cycles arrest will be there. So these oncogenes induce senescence will be started, and the cells will not divide ahead. So you see there will be no uncontrolled proliferation. So when we are talking about the tumor suppressor genes, what are other types of tumor suppressor genes? So I just have taken a list out of a research article, out of a review article actually, which beautifully summarizes the tumor suppressor genes in terms involved in human neoplasm. When we are talking about the location in the at the cell surface, there is TGF beta receptor, tumor growth factor beta receptor, which is responsible for growth inhibition, and it has been shown that any aberration in TGF beta receptor has been shown to be present in colon carcinoma. When we are talking about plasma membrane, then NF1 or NF2 protein, they induces cytoskeletal stability. They're responsible for cytoskeletal stability. Any change or any mutation in these two genes have been shown to induce either neuroblastoma or meningioma. So, like these, the list is although short, but these proteins perform very critical function in terms of cell cycle control, whether it is P10 like PI3 kinase signaling transduction, which is responsible, or its aberration is responsible actually for induction of prostate cancer in somatic cells. So these are because of the somatic mutations. When we are talking about the inherited mutation, it falls in the category of Cowden syndrome. Cowden syndrome. Then when we are talking about the retinoblastoma, which is present inside the nucleus because the cell cycle is going with the nucleus part, it is responsible for regulation of cell cycle. Likewise, P53 is responsible for cell cycle arrest or apoptosis when there is a DNA damage. If any mutation, any ionizing radiations, or any kind of exposure any DNA damage, that suppose the strand breaks or the single strand breaks, this P53 is getting activated, and it is responsible for putting that cell towards programmed cell death by inducing pro-apoptotic proteins, the Bex family protein. So you see, any change in these two proteins will result to aberrant functioning of the cell, and that aberrant functioning in terms of cancer is known to cause retinoblastoma or osteosarcoma or colon cancer, lung cancer, breast cancer, and almost multiple types of cancers have been shown to have aberrant P53. That's why it's one of the key regulator proteins which has gone for very in-depth investigation. It's gone for very in-depth investigation, and the inherited mutations that has been shown to be associated with the tumor. Those mutations can lead to Li-Fraumeni syndrome. The aberrant P53, the mutated P53, leads to Li-Fraumeni syndrome. And other genes which are gaining the limelight, which are the DNA repair genes, are BRCA1 and BRCA2. These are known to be involved in DNA repair. So if there is any kind of mutation in these particular genes, the DNA repair system is faulty. If DNA repair system is faulty, then there is multiple onset of carcinoma, and these two genes have been technically identified in breast cancer. So you see, there are multiple sets of tumor suppressor genes, out of which the genes which we are talking about present in the nucleus are retinoblastoma, RB1 and P53. So when we are talking about tumor suppressor genes, the question is whether these genes are actually suppressing the tumor. Then that's why I told you earlier that that's a misnomer. It never directly suppresses the tumor, but if any change or any aberration happens in these genes, then that might lead to cancer. So you see, we are holding a ear not from this side, but we are trying to hold it from the other side. Why? Because I have given a nomenclature as tumor suppressor gene which will suppress the tumor. It's simple, but no. This is a gene which is required for normal functioning. If any change happens to this gene where its functions become impaired, then the normal response will be transformed to carcinogenesis. And that is why we are calling them as tumor suppressor gene, because loss of function may result in generation of tumor. Okay, so this is a kind of a misnomer. So when we are talking about the pathways that by mechanism by which this tumor suppressor gene is going, that it controls over the mitosis, and it also controls over the other biochemical processes that a cell requires for proper growth and differentiation. When we are talking about the P53, see, once the P53 gets activated, now it can get activated by many kind of insults. It can be a DNA damage insult if there is any kind of DNA damage, or it's a kind of an oncogenic signaling. The oncogenes are expressing like RAS protein is getting activated. It is getting activated towards its GTP. Then it will activate the P53 transcription factor. Or there is any kind of nutrient deprivation or a condition of less oxygen concentration called hypoxia, or there is a ribosomal stress, or there is a ionizing radiation exposure. The ionizing radiation exposure will in turn cause the DNA damage, thereby activating the P53. So you see, once the P53 transcription factor or the P53 gene is activated, it will give rise to P53 protein, and the P53 gene is in front of you. It has given the expression in terms of P53 protein, which is called as transcription factor also. This particular protein will go inside the nucleus and will bind to P53 response element, which is present inside the DNA of human. Now, after binding, it will promote the transcription and expression of its downstream genes. And what are those genes or proteins? The proteins which are formed after the expression of its downstream proteins are involved in multiple cellular pathways. What pathways? Pathways like apoptosis. P53 is known to induce PUMA to induce the apoptosis or the back family proteins. The pro-apoptotic proteins are induced in response to P53. It is known to control the cell cycle as checkpoint. It is known to be involved in activating the pathways of DNA repair or in normal cellular metabolism or in maintaining the redox equilibrium, the maintaining the cellular redox homeostasis. It is known to play a very crucial role. Then in whether we are talking about any kind of reprogramming or differentiation or response of tumor microenvironment, this P53 molecule is a very key transcription factor which is known to play the diversified or pleiotropic roles inside a cell. So you see, if somehow this particular P53 is getting aberrant, then there is no one to check whether there is DNA damage or not. If there is no one to check the DNA damage, then this particular cell cycle will keep on going, and eventually you will see that the damaged cell is getting accumulated and has gained the ability of uncontrolled growth. So when we are talking about the P53, it induces the protein called as puma. This puma protein results in increase of apoptosis, thereby results in tumor suppression. So this particular slide is predicting how this P53 is called as tumor suppressor gene. So when the P53 is activated, it is present in ample. The gene is intact. What is happening? It is giving rise to P21. Now this P21 increases the cell survival. Also, it increases the senescence, thereby giving the cell ample time to repair its damage. It induces cell senescence. It induces a cell cycle arrest. It is the protein which is released in response to the oncogene expression. Whenever there is an oncogene expression inside a cell, the P53 is getting activated. After getting activated, it results in the formation of P21 protein, and this P21 protein in turn induces cell cycle arrest, that is senescence, and thereby giving cell ample time to first repair that particular damage, or the oncogene expression should be shut down, and then only cell will be coming out of that senescence. Then if it's not possible, let's go for apoptosis. Let us go for the programmed cell death of that particular damaged cell, and that is induced by puma. Or let us increase the autophagy, which will again go for tumor suppression. Or let us inhibit the mTOR pathway, which is very much required in terms of tumor progression. So if you are inhibiting mTOR, you are in turn inhibiting the tumor progression. So this is how a kind of a summary, for the pleiotropic effects or pleiotropic pathways of P53. But if you require to study these pathways in detail, you can directly go to the database. You can directly go to a Swiss prot. You can see there the biological function, the molecular functions, or you can go to the Keck database and you can see the complete signaling network that this P53 protein is involved in. It's a very beautiful representation where there's a collective chart that will be available for you. So you see, once what we have established till now is that P53 is very crucial in order for a normal cellular response. So whenever there is a carcinogenic insult or a kind of a mutagen that is being exposed to the cell, or when we are talking about any kind of ionizing radiation exposure, there are certain damages to the cell, whether in terms of the DNA damage, there are DNA damages, and that's the root cause of these particular carcinogenic insults. So when we are talking about the normal cell, that normal cell will in response to these insults will inculcate the damages. Of course, the damages will be there. So, fine. Then the condition of hypoxia will be there with this particular condition. This will activate the P53. The P53 will go and bind to the portion where the DNA got damaged, and then it will start the transcription of its target gene. It will start the transcription of P53 dependent gene. the p53 will go and bind to p53 response element and will result in the formation of p21 protein. This p21 protein is actually the cyclin dependent kinase inhibitor. If it inhibits the CDK, the cyclin dependent kinase, then it will shut down the progression of cell cycle from G1 to S. And if there is no progression from G1 to S, then the cell is at arrest state, and that is G1 arrest, and that is called as senescence. So when it is giving a G1 arrest, then it is giving a ample time to that particular cell to repair that damage. Then the DNA repair pathways will be getting activated and they will be repairing that particular damage first of all, and then only that particular normal cell will be allowed to progress ahead. And then there is another set of protein, GAD45, which is responsible again for DNA repair. But what if the repair fails? What is the fate of that arrested cell? Then the p53 simultaneously induces pro-apoptotic proteins family called as Bax family by a Tumor line, and that particular protein is responsible for inducing the apoptosis in the arrested cell, or in the defected cell. If there is no chance of DNA damage repair, then that particular infected cell needs to be cleared out, and that clearing out is regulated in a controlled manner or a programmed manner called as apoptosis, mediated by pro-apoptotic proteins. But what happens to the right side axis? What right side axis? It's not a normal cell. No, it is a mutated cell where the p53 is mutated, where the p53 has lost its function. That mutation in the p53 can happen by any chance, right? It can happen spontaneously also. It can happen by any kind of mutagenic insult also, or it can happen by virus infection also. The persistent viral infection, we'll see. So what I'm trying to say is that the cell is mutated here. In response to these stimuli, there will be DNA damage, and there will be no one to sense this DNA damage. The p53 dependent genes will now not be activated. They will not be activated. There will be no cell cycle arrest. There will be no DNA repair, and the mutant cell is now living free. This cell is very happy. Now this cell, if there is no checkpoint, it's just like a highway road. If there is no toll tax or there is no toll gate, you will be going ahead with the 120 km/h speed. But if there is toll tax, if there is toll gate, if there are certain PCR vans which are monitoring your continuous speed, then you will be going at a very slow speed at defined of 80 km/h. The same scenario is here. When the mutant cell sees that there is no one to check, no repair pathway, then why to waste time? Let's proliferate at a very exponential rate, which leads to expansion, as well as additional mutations will keep on growing, which will give rise to malignant tumors. And this is how the pathogenesis of cancer occurs in response to p53 gene. So you see, we have clearly established that what is the mechanism of p53 gene. What are the consequences if this particular p53 protein is aberrant? So moving ahead, let us talk about the protein products of these tumor suppressor genes. Just not talking about the specific products, but the specific pathways in which these products are involved. See, the tumor suppressor genes or the p53 genes products can be another transcription factor, which is again enhancing certain kind of proteins, or it can be a cell surface receptor. It is definitely a part of regulating the apoptosis or the cell survival of the growth or cell cycle control, or in response to DNA damage. These proteins are technically seen. So what are the tumor suppressor genes? Apart from retinoblastoma and p53, there are certain lineages or the certain genes that are actually being shown to be very potent, like WT1 gene, Wilms tumor gene. It's again a very potent tumor suppressor gene, or BCL2 family or BCL2 gene family. BCL2 gene family got its name by B cell lymphoma. If this gene is aberrant, then of course B cell lymphoma sets in. So there are certain set of protein. When we have discussed the complete biology or the complete process of that particular p53, let us focus now on retinoblastoma, the pathogenesis of retinoblastoma. The retinoblastoma is a cancer caused by what? Caused by aberrant RB protein that we discussed. That's why the name transit from RB to retinoblastoma. Fine. That's so. This the picture is depicting your cell cycle, right? G1S, G2 and M, and then there is a G naught, which is a cell cycle arrest state. There is a cell cycle arrest state, and then we have seen that cyclin D along with CDK4 is responsible for phosphorylating the retinoblastoma. So in at initial stage, when we're talking about at initial state, the retinoblastoma, the RB protein is hypophosphorylated. The phosphorylation level is very low. In response to the transition from G1 to S, in response to a transition from G1 to S, what is happening? What is happening? It will active. It will cause hyperphosphorylation of retinoblastoma. The activation of these kinases, the CDK4 and CDK2, will result in hyperphosphorylation of this retinoblastoma. So what are the things that can activate these particular kinases, like growth factors, the epidermal growth factors, or the stimulus, which are saying that yes, growth is required, please go for the cell cycle, and then this particular status will the phosphorylation status will change for the retinoblastoma. Whereas the growth inhibitors like P53, the checkpoint inhibitors like P53, the TGF beta, the tumor suppressor gene will actually stimulate the CDK inhibitors like INK4A, CYP, KIP, which will in turn inhibit these kinases. It will inhibit the cyclin CDK association, thereby inhibiting the hyperphosphorylation state of retinoblastoma. So this is how the regulation is happening between the growth factors and the inhibitors. And that is your actual checkpoint. And that is your actual checkpoint. If growth is required in response to growth factor, this will be activated. If growth is not required in response to growth inhibitor, this will not be activated. But what is happening when we are talking about virus insult? See, oncogenic viruses, what they are doing. Oncogenic viruses, what they are doing. They will go and bind. They will go and bind to these. They will not the viruses actually. In fact, the viral proteins will be responsible for this particular aberration, and they will go. They will bind to this particular hyperphosphorylated state. Once the hyperphosphorylated state is aberrant, it will not be able to cause the checkpoint that is required. It will not be able to form the checkpoint, and that's why the cell will be easily transit. See, if this particular retinoblastoma is aberrant, then the effect of CDK will be happening on nothing. The growth inhibitors will not be able to cause any change on retinoblastoma, thereby there will be no module to regulate the G1 to S transition. Fine. And that is why this protein is very important. What is RB protein? The RB protein is a protein which is actually regulating the cell cycle. It's a phosphoprotein which is getting phosphorylated. Once it is getting phosphorylated, the active phase, what we are calling, what is the active phase here in the last slide? See, the active phase is always hypophosphorylated state. The hyper, where the phosphorylation is very high, that is inactivated state. So once it is getting inactivated, then there is no function. So the active hyperphosphorylated state is always in the quiescent cell, whereas the inactive state is responsible for the transition. So you need to inactivate the retinoblastoma protein. In order for a cell to go from G1 to S, and that is the thing. The viral protein inactivates the retinoblastoma. Thereby it loses the power to induce the arrest state or the quiescent state, and thereby loses the checkpoint stability. Right. So what it is doing, that it regulates the advancement of the cells from G1 to S phase. That what we have understood, in response to growth inhibitors, whether it is TGF beta or the other cytokines that in turn results in upregulation of CDK inhibitors. What they are doing in itself, they are preventing the hyperphosphorylation. They don't want the retinoblastoma to get inactivated. That is the main function of those growth inhibitors. So when we had understood that the indirect mechanism and the tumor suppression part, what is the direct mechanism? What would be the direct mechanism? Direct mechanism is such where the infected cell is actually becoming the malignant cell. How it is happening? It is happening simply because the virus which has infected the cell, it is inducing the oncogenic expression, or it has directly caused certain genotoxicity inside the cell, which has resulted in the transformation of normal cell to the oncogenic cell. So the DNA viruses that are dependent on the host DNA polymerase, of course, for the replication, they form certain gene products that promotes the cell cycle. That promotes cell cycle. There is, the normal cell cycle time span is have now become impaired. So there will be continuous cell cycle, continuous cell proliferation will be there. That is the typical mechanism. Now, why we are calling it as direct? See, because the oncogenic effect mediated by these viruses is directly on the p53 or retinoblastoma protein. That's why there is direct aberration of tumor suppressor gene in these by induced by these viruses. So when we are talking about the viral proteins, why we have mentioned the direct hit and run mechanism. When we are talking about direct mechanism, we are calling it as hit and run mechanism. Now, what is hit and run mechanism? See, when we are talking about the viral gene product, it may preserve your viability, or it may promote the cell growth. But what is happening in a nutshell? What is happening? See, there is a genetic damage. There is a genetic damage whenever there is a early phase of tumor development. Whenever there is an early phase of tumor development, there is a genetic damage. So when we are talking about the number of genetic damage, whether a single genetic damage is sufficient enough to be substantiated for a carcinogenesis phenomenon? No. Yes, but the onset is there because of that genetic damage. Because of that genetic damage in the early phase of tumor development, the cell, it allows the cell to accumulate multiple damages. It allows the cell, or you should say, not the normal cell, it allows the precancerous cell to accumulate enough damages or enough genetic alterations which will eventually allow cell to grow rapidly and survive, fine. And when the viral gene, or you say when the cell is affected by the virus, that particular viral gene has expressed certain proteins. Now these proteins will be responsible for suppressing the immune responses. So suppose the infected cell has now become aberrant. There is certain genetic alteration which has transformed this cell into a tumor cell. Then it was the responsibility of this immune system to identify this aberration and clear the cell. And the role of the checkpoint was to identify that aberration and put this cell in resting state so that it can be repaired or removed by apoptosis. But now, because since the viral proteins are present here, they are suppressing the immune responses, and they have also altered the tumor suppressor gene. This cell, which has become now aberrant because of genetic alteration, now is superior. Why? Because it has another associate, the viral gene, which is doing all the functions to increase the survivability of this particular cell. The cellular viability, the viability of this infected transformed cell now increases because there is no immune response, there is no checkpoint. And eventually what happens, this gives this particular cell ample time